So I think it's going. Yep. So hello, everyone. We are happy to have you here to enjoy with us one more session of our monthly seminars of the International Academic Network. So our initiative with this seminar is trying to keep our community engaged with the research and the outreach topics related to cacti. So to those that didn't receive the link, please make sure that you have subscribed our forum. I will put the link in the Zoom chat. If you have subscribed before, you don't need to do it again, but some people haven't not received the link. So, and you can also find more about the further seminars in your Twitter account. It's Cactaceae_net. I will also post in the Zoom chat. Today we are welcoming Dr. Fernando Fraco. Dr. Fernando is professor at the Universidade Federal de São Carlos in Sorocaba City, São Paulo State. And the goal of his research is to investigate the evolutionary process underlying species diversification, especially those found in xeric and open areas in the neotropical region. To this end, he and his group have been exploring different aspects of evolutionary genetics of cacti, including projects related to population genetics, biogeography, phylogenomics, and also transcriptomics data. So Fernando, we thank you for the opportunity to have your talk today, and it's up to you now.
Thanks for the introduction, Matthias. Hi, everyone. First, I'd like to thank the organizers of the seminars, Matthias, Tanya, Gustavo, Eddie, and everybody involved. I believe somehow these seminars are keeping we connected each other. So I'm very glad to be here, and I thank you for organizing this event. Before I start, I have to say I'm not a fluent English speaker. I have some notes with me here to guide my presentation. I hope you can follow the main messages and that we have time for discussion in the end. The title of my talk is a question for which we know the answer already. Not always a biogeographical movement should be accomplished of niche evolution. Then my aim here is not to show too much results for you, but to discuss with some ways to distinguish between a simple range expansion from a biome shift. I will show what we did a little bit and what we plan to do to address this problem. So to begin, biogeographic movements or geographic transitions, we can understand about a movement of species from one location to another location by dispersive events. And these movements are crucial to understanding the assembly of regional biota. A common way to describe these geographic transitions is using biogeographical reconstruction based on phylogenies. So to do this, we define some operational geographic areas based on researcher experience or some map of biomes or domains, and then assign the taxa in these operational geographic areas, and then perform the statistical analysis. For example, in this paper of 2018, Antonelli did a lot of biogeographical reconstruction for many taxa and identified that Amazon is a primary source of dispersion for many taxa, okay? But these geographical movements, including these described in this paper, are not necessarily a biome shift. Many times we use domains as synonymous of biome, but biome have a quite controversial definition. I'm not to enter in details, but there are many papers discussing this issue. But important aspect of biome definition is that biome definition is a azonal concept. That means that bioregions or domains cannot be properly defined always as biome. Okay, we have savanna in Africa, but we also have savanna in South America, Australia, and another places. So yes, is a seasonal concept. And biomes are vegetation units defined by functionally similar plant groups. This is truly important. And similar environmental conditions. So taking these concepts, highlighting functional aspects of the vegetation units, Donoghue Edwards discussed important aspects and concepts for the interpretation of biome shifts. For these authors, biomes should incorporate units with similar niches and organisms with functional attributes shaped by natural selection. So in this view, very summarized way, biome shift can be approximated of the concept of habitat shift, which can be approximated of the concept of niche evolution. So this is crucial to understand biome shift in the biological sense. So two scenarios can occur during biogeographical movements. We can have niche conservatism, which by far is the most common process during the expansion from the domain 1 to the domain 2. There is an overlap in the niche dimension, and then we can describe this as a simple range expansion, but not necessarily accomplished with evolution of niche. And in a second scenario, when the geographic expansion can be associated with habitat shift or biome shift. In this second case, we have no niche overlap, and it is possible to describe as a biome shift. Okay, but there are practical challenges here. For example, how to measure the niche. This is very hard. Niche is a multidimensional category, so it's very hard to define it properly, but it's possible to measure some components of the niche. For example, climate. And by the association of the niche information that you have with phylogenies, we can moving forward in understand the biome transition. For example, in this this figure here, we have two species co-distributed in eutrophics, the blue one and the green one, that occupy the same climatic niche, the same conditions of temperature and precipitation. On the other side, the third species, the black one, occupies a new distribution area, exploring a new climatic niche. This would be characterized, sensu Donoghue and Edwards, as an invasion with niche evolution, or a real biome shift. So let's go to cacti that truly matter for us. We have been exploring a lot of biogeographical reconstruction and described a lot of interesting biogeographical movements in cactus family, in both family level and the species level phylogenies. Here, I'm only showing two very influential papers in two taxonomic scales. And nicely, many of these papers also brings ancestral trait reconstruction, which give us some idea of key traits involved in diversification events. So, but, however, as you know, cacti has other interesting aspects not properly addressed in many biogeographical reconstructions. For example, I'm projecting here a very nice paper of Aquino et al., published last year, highlighting the phylogenetic signal of some abiotic traits, many climatic variables that the authors got from word claim, and also some soil variables that the authors got from a Mexican database, very detailed database that allows the authors map along the phylogeny. Aquino et al. Did a lot of analysis, but I'm showing here two ancestral character reconstruction of two abiotic variables, and we can see a clear pattern of change in the clade of genus Epithelantha. So the authors argue that there is some abiotic variables which has phylogenetic signal in this genus. This is a very nice result. We know that this kind of pattern is very common in cacti, and therefore, I believe we have a great opportunity to study the process underlying niche evolution using cacti as a biological model. So I want to address if during the biogeographical movements, what kind of pattern is more frequently observed? The cactus are tracking more or less similar habitats along the expansion, or they are adapting to different biomes. For example, this is the distribution of Brazilian eucalypt forest. This is a humid biome. The core vegetation is very dense and closed, and the cacti in this biome grows, the most of cacti grows in parts of xeric vegetation like this. So to what extent this environment is different from a savanna or a typical dry forest? That's the main question I'm interested in the new project. So to answer these questions, we need to address analysis about niche overlapping, try to investigate if there are enable traits, traits that allows or drives niche evolution, or if there are some signatures of natural selection. Of course, you can say that there are a lot of alternative ways to address these questions. I agree with that. I will show how we are dealing with this in genus Cereus, okay? But we can discuss at the end alternative ways to do the same thing. So very quickly, the genus Cereus has about 25 species with wide distribution in South America and occur in different climatic and soil conditions. So I believe it is an attractive model to study biome shifts. We explore biome shifts in a paper led by Dr. Danilo do Amaral, published last year. In this work, Danilo reused some RADSEC data to perform many types of biogeographical analysis here. And it is important to say that our main approach, or our first approach here, was to discuss only the geographical movements, only to reconstruct the ancestral geographic areas and the dispersal events and the vicariance events. But during the peer review, we were challenged by one reviewer about what we were calling biome shift, or what we were calling biome. So these questions greatly improve not only the work, but also how you see our plants now. Okay, so this a little bit change of approach allowing us to discuss questions like the geographic range movements were followed by niche evolution, or there are phenotypic traits that could be involved in biome shifts, and I will show very quickly some results. This is the biogeographical reconstruction based on these areas here, and we identified many biogeographical movements along South America, represented by the arrows in the phylogeny. Most of them rooted in the Cerrado domain. Cerrado is a savanna-like vegetation in the middle of Brazil and in the middle of South America. And most of the spatial events was out of Cerrado. Okay, I will highlight only 3 clades here as examples, which I will explore along the presentation. Clade D is a clade with a biogeographical history of colonization from Cerrado to Caatinga. Clade A2, a dispersal from Cerrado to Brazilian Atlantic Forest. And clade we call clade Jamacaru, also a dispersal from Cerrado to Caatinga. I will take these 3 clades to illustrate the main thematic of this talk. Besides biogeographical reconstruction, we also did analysis of biome affinities reconstruction. This is a kind of ancestral character reconstruction, but using biomes categories as the variables mapped along the phylogeny. We define 2 categories of biomes. The main difference between them is that in the second, we split seasonally tropical dry forest by their regional names. And in the first, we consider seasonally tropical dry forest as a single azonal biome. Okay, and the other biomes was neotropical savanna, restinga forest, Atlantic Inselbergs, Patagonia, and Chaco. In this analysis, we saw a lot of transitions along the phylogeny too, but I will highlight those 3 clades again. If we highlight these 3 clades, we can see a state change in the tree. For example, here, a state change from savanna biome to seasonally tropical dry forest. Here, a transition from seasonally tropical dry forest to restinga forest, and to restinga, and then to Inselbergs. And here, in the third, some transitions inside the seasonally tropical dry forest, from one type of seasonally tropical dry forest to another type of seasonally tropical dry forest. Okay, so those 3 clades could be involved in a real biome shift. However, when we perform a niche overlap analysis comparing each clade pair to pair, analysis not showed here, together with the ancestral reconstruction of climatic and soil variables, we observe shifts in the climatic niche of this clade D here is very distinct of the ancestral, and in these other clades, clade Jamacaru, also a change here in the species Cereo Jamacaru, but not in the clade A2, inside the Brazilian Atlantic Forest. So we believe this 2 has all characteristics related to biome shifts after a biogeographical movement, but this clade, no. Okay, because the niche overlap with the ancestral niche. We also did ancestral character reconstruction based on 36 traits. And we found some traits with phylogenetic signal along the phylogeny. But again, regarding our target clades here, we found state changes in both clades. In clade Jamacaru, we got state change in the rib margin, and in clade D, we got state change in several traits. So this clades has evidences of biome shifts, has evidences of state changes in phenotypes, and the question now is if these changes of states represents enable traits. In other words, these traits were important to the biome shift or not. Okay, so as preliminary conclusions, we can say that some biogeographical movements in cereals are simple range expansion, for example, in clade A2. Some biogeographical transitions or geographical transitions in genus cereals may be considered biome shifts in the sense of habitat shifts, for example, clade D and clade Jamacaru. And also there are some K-enabled traits that could be involved in biome shifts in genus cereals. Our focus now is to understand the process of adaptation during the biome shifts in the genus cereals. So for this end, we started a new project trying to integrate phylogenetic, genetic, and functional attributes. So this integrative approach will be allowed by our phylogeny, but also integrating low coverage genomes, transcriptomes, and ecological data, phenotypes, abiotic variables. We are trying to also to get chemical compounds of cacti to try to resolve this puzzle about it, to understand the process of adaptation in cereals. It's important to say this is also the approach to be used in a larger project involving a network of researchers from Brazil and United States. The United States team is led by Scott Edwards, and the Brazilian team is led by Vera Soferini from Unicamp, and me and Professor Evano Moraes. He's here with us. We are in charge to investigate the cacti with this main ideas to investigate the process underlying biome shifts. Okay, there are a lot of other organisms which will be analyzed in this project: reptiles, birds, insects, and so on. And the idea is to compare the data in the end of the project to discover similar process. So please stay tuned in this project. Very soon we will have a web page to better spread the message. Back to cereals, we will investigate the clade in this new project, the clade D and the clade Jamacaru. In common, these clades are associated to biogeographical movements from Cerrado domain to Caatinga domain. And in common, this clade has also the evidences of a real biome shift. Okay, as I try to show you. With these two players, we intended to respond to this kind of questions. Are the common signatures of natural selection in independent transitions from the Cerrado to Caatinga, experienced by the genus cereals? Also, there is some particularities in both clades regarding to a particular environment that they occupies. And to start this project, we did a field trip. We initiated this project during the COVID pandemic, but recently we could finally go to the field. We did a very cool field trip exploring a lot of region of Eastern Brazil and performing sampling of different types. We got roots for DNA extraction, material frozen for RNA extraction, tissues frozen also for chemical analysis, and soil from the roots for macro and micronutrient analysis. So in this field trip, we also explored different habitats where our target clades grow. I will show very quickly some pictures here. We collect in campo rupestre vegetation, is a kind of rock outcrop vegetation, a kind of savanna above the rock outcrop. It's not properly a savanna, but it's a savanna-like vegetation where grows some cacti, for example, outgroup of Cypocereus brade. We sampling ecotomies of Cerrado Caatinga. Here, this location is in sand soils, a place where we have elements from Caatinga, but also elements from Cerrado. And here is the species from clade D, Cereus mirabella. More campo rupestre. This is Cereus mirabella also, but now growing in rock outcrops. We sampling sand phases of Caatinga, called in Portuguese Mata de Carrasco, where we collect Cereus albicaulis, Cereus jamacaru, collecting Cereus, sampling Cereus jamacaru also in granite derivative soils. I don't know if you can see, but there is a rock outcrop here. This rock outcrop is derivative of granite. Dry forest growing in granite outcrops, limestones. This is a very interesting environment. This type of environment may be surrounded by savanna, but above the granite outcrop grows dry forest, so works like island of dry forest vegetation in ecotomies of Cerrado savanna and in Cerrado. More dry forest here. And okay, as a methodological approach, we will follow a PhyloGeoWAS approach in this project, using these different databases, as I already say, low coverage genomes, transcriptomes, metabolomes, and ecological data, in order to see if the transition between these environments could be explained by natural selection, for example. So we can compare different scenarios within this approach. For example, we can compare the samples from savanna versus the samples from Caatinga, a seasonally tropical dry forest. We can compare two seasonally tropical dry forests, one growing in limestone and another growing in sand phases of Cerrado, of Caatinga. We can compare another scenarios. For example, we have a plant, a clade that occurs in Brazil Atlantic forest, one clade in the coast, in another clade in the island. So we can compare these two sampling too, and etc. We will refine this along the project. But the main aim is to understand or to find nucleotide sites, genes, metabolic pathways, or chemical compounds which help us to explain the biome shifts, okay? So please, if you enjoy this approach, contact us. We have opportunities for post-graduation or postdoctoral projects. For those from outside Brazil, we have programs to receive foreign people. And more importantly, we need human resources for helping us to achieve these aims. As final remarks, I would like to highlight that cactus, as you know, has a series of adaptations to survive in many diverse habitats regarding to abiotic and biotic conditions. We know that the xeric habitats or the dry lands along the distribution of cacti are very different. So is as I said before, I believe is a nice opportunity to not only understand the geographical movement, but also try to understand what changed in the cacti to occupy a new niche. Cactal biogeography requires efforts towards an integrative approach, a trait-based approach. So we need to refine of data sets of phenotypes. We need to integrate ecological data as much we can, and refine all kinds of data sets to improve our analysis. And of course, to do this, we needed to try to unify databases, for example, databases of phenotypes or databases of soil. We don't have a unified data set, and then would be very helpful to do this kind of work, or even necessary to do this kind of work. And of course, always collaborative efforts to integrate cactus researcher. We know much of us has different expertise, and putting all this together will be very good, not only for us, but also to understand this very nice family. So I'd like to thank Evandro Moraes and all people of our lab, Lagevol, for constant collaboration, the taxonomists, Daniela Zapp and Nigel Taylor, that also contribute a lot for our projects, the funding agencies and institutions, and of course, you for listening to me, and thank you. I hope you will have time for some questions. Matias?
Yeah, thank you, Fernando. That was amazing and so much exciting talk. Nice. A lot of different topics that we can discuss a little bit more now. So everyone that may have some question, please, you can use the chat, or even you can mute yourself to ask directly to Fernando or anyone else. You can also use directions from Zoom to hands. Oh, Lucas already hands your hand. Go ahead.
Sorry, I'm jumping the gun here. No, I just want to say, Fernando, really great talk. I've been reading your papers as they come out from this project. You know, it's a really, really nice project. I really love the integrative approach you guys are taking as well. So I had one question regarding, you know, either habitat or biome shifts and biogeographic shifts just in general. Do you see any major patterns and morphological change in the genus Cereus when you see these major shifts in you know, either biomes or even major biogeographical changes?
You mean in the phenotypes?
Yeah, in the phenotypes.
Yeah, some clades we got signal of biome shift and then signal of phenotype shift. But the question is if always the question is if the change in the phenotypes what is a cause or a consequence of the evolutionary process. So I believe now integrating some genes, some transcriptomes, maybe we could find a sign of natural selection in these genes. And if we have luck, the gene is involved in trait expression.
Yeah, great.
Yeah.
Thanks. Yeah. And I think that's super important because, you know, there are definitely these, I think, you know, and even in other groups of cacti, there are these what appear to be very clear shifts in morphology when you shift into different habitat types and or biomes, et cetera. But we often don't have this underlying genetic data to further test that. So, so I'll be really excited to see what you guys find there.
Yeah, thank you. If you have more questions in Spanish or Portuguese, feel free to.
I got curious because when you show your biogeobeers reconstruction for cereals, you have like Cerrado as the ancestral area. But when you use like the biome, like a concept, you find like seasonal dry tropical forest. So this is kind of, I don't know, blow my mind. Like, oh, and it's funny to find like Cerrado as a putative ancestral area for cereals because I am not expert in cereals, but to the extent species diversity and even Cerrado is not I don't know, nice place to cacti. It's a fire regime environment, so it's kind of I don't know. I would like to know your thoughts about.
Yeah, I agree. We also had the same questions about Cerrado. Cerrado is has a big extension, a big geographic extension, but there are different types of environments, and in most of cacti growing in Cerrado grows in rocky outcrops.
Yeah.
Because the cacti are not fire prone, and Cerrado is a fire prone biome. So there is some diversity of cereals in the Cerrado, especially in northern part. Now, but this area is very poorly known yet. Some months ago, Gerardo's Alstons find a new species of cereals in northern Cerrado. So I believe there is a lot of places to explore yet in the Cerrado for cacti. But regarding the analysis, yeah, you are right. When we change operational geographic units in Biogeographical, we can change all the results and even the model selected. When we use the domains, the best model, including jumping dispersion. So it's a model that somehow take a lot of weight in dispersal events. But when you try another scenarios, even the model changes. We try a lot. Danilo is here, did 3 or 4 scenarios and changes a lot. But this is a limitation of the methodology and the how artificial is we create operational geographic units. Because of this, we disentangle the analysis, one geographic using the operational geographic areas that made more sense for us, and other based on ancestral character reconstruction for the biomes. So even so, we did 2 analyses, considering seasonally tropical dry forest as a whole and splitting it because this changes. But yeah, it's a very nice question. It's blowing our mind too.
I have seen similar discussion with the Caatinga biome, like the Sensulato Caatinga and Caatinga Sensu-Strito, because in Caatinga Sensulato you can find, I know, Atlantic forest and Campos rupestres and different kind of biomes. So maybe for Cerrado, we can also think about similar approach to delimit the vegetation units, and then make more sense to, because when I heard Cerrado, I think in a savanna-like with fire-prone ecosystem, but the cacti are in the rocky outcrops in Cerrado, so it's,
even in Cerrado, the limestones includes dry forest. Is inside the Cerrado domain, but is biome similar to Caatinga. It's very curious.
I don't know if someone else have questions to Fernando or about the topic.
I don't know if I talk a lot or very fast.
There is a comment in the chat box.
Oh, sure. Suggestion.
Yeah, we had a comment about the cacti growers, so.
Nice. Thank you for the comment, Martin.
Can I ask one more question? Do you think there will be like important associated microorganisms that could be shaping this shift between biomes in Sirius?
That's another really good question. We don't know, but we know that in some cacti there is microorganisms, endophytic microorganisms that could have some role in the occupation of an environment. But unfortunately, we haven't this information for Sirius.
Right. And would you be interested in introducing like microbiomes analysis in your approach, in your integrative approach?
Yeah, would be very good for us to know some analysis like this. There is some works that do the microbiota of the soil associated with the roots, the rhizomes, and also endophytic microorganisms. Would be very good information for us, for sure.
Okay, thank you.
Thank you, everybody. The chat, Evandro, Alton, Monique, Adriano, and everyone. Thank you very much.
Yeah, we have many people congratulating you. Great talk.
So, okay, there is a question of Monica here. about the price cereals. The tree was rooted with price cereals. What we can tell about this? Yeah, the question is, what can you tell about its relationships to biogeography of cereals? Yeah, in 2020, we published the cereals phylogeny based on RADSEC data, and price cereals was a sister clade, or I believe is a sister clade together with the other species of a clade composed by cypocereals and cereals. And then here we consider cypocereals as an in-group. So we choose price cereals as an out-group just for the biogeographical reconstruction. But of course, the out-group will be important to establish the evolutionary relationships, but for the biogeographical reconstruction, that is not has no influence, because we stopped our interpretation in the node of cypocereals and cereals. So the biogeographical reconstruction came from the tips to the internal nodes. Then I believe there is no influence. I don't know if I answered. Thank you, Monica, for the question. Someone else?
Okay, Fernando, sorry. I do have one last question, actually.
Okay.
So I'm wondering, so it seems super curious, you know, obviously cereals seems to be a quite young clade, right? Relatively rapid radiation, perhaps. I'm wondering what you guys might know with regard to chromosome number across cereals. Do you have a really good idea of all the species, whether or not there's any polyploidy in the mix that might further sort of obscure potential reticulation events or something like that in the phylogeny?
Yeah, about chromosome number, all information that we have, the species are diploid. For those species which have cytological plates. And based on variability in microsets or even RADSEC data, all the data suggest diploidy for all species. Regarding reticulated evolution, in the in the paper of the phylogeny, we found some integration signals, but we really not explored a lot reticulate evolution with many different analyses.
Great, thanks. No, yeah, that's yeah, it's super fascinating. And you know, I guess I would say that that's you know, even better to have a group like that as a model group if they're all diploids. That helps a lot.
yeah, I agree.
Yeah, excellent.
Yeah, all are diploids until we find a polyploid there.
Right. Yeah, that's definitely a possibility.
Okay, guys, I know someone else have questions. Then we can enjoy the last time to finish this meeting and wait for the next one.
Okay. Thank you, everybody. for
Thank
the
you so much.
discussion.
Okay.
See you.
Thank you everyone for coming. It was really nice. Thank you, Fernando, and everybody.
Thank you, guys.
See you soon.
See you
Okay.
soon.
Take care. Bye-bye.
Bye.